Welcome to Oncology Morning Commute. What's next in using PARP inhibitors for patients with ovarian cancer? Morning Commute is developed by Projects and Knowledge powered by Kaplan and is part of a continuing medical education series. This independent CME-CE activity is supported by an educational grant from GlaxoSmithKline and Merck Sharp and Dohme Corporation. In this final podcast of our three-part series, Dr. Robert Coleman and Dr. Kathleen Moore look at some of the latest findings on PARP inhibitors, as well as what is on the horizon. Is there synergy between PARP inhibitors and other agents? Will double or triple combination therapies be the next step in ovarian cancer treatment? They also take a look at disparities of care in ovarian cancer and how clinical trials are addressing this. Information about the faculty and disclosures can be found at morningcommutepodcast.com/ovarian cancer 3. You can use this link to receive your credit and evaluate this program. The URL can also be accessed in the episode notes. Dr. Coleman is the Chief Scientific Officer at U.S. Oncology Research at Texas Oncology, Shenandoah, Texas. Dr. Moore is the Associate Director of Clinical Research in the Department of Gynecologic Oncology at the Stevenson Cancer Center in Oklahoma City, Oklahoma. I am your host, Candace Hoffman. Dr. Coleman will begin our discussion.
Well, good morning, everybody. Welcome back to podcast number 3 of 3, discussing PARP inhibitors in patients with ovarian cancer. Of course, joining me today is my good friend and colleague, Dr. Katie Moore at the Stevenson Cancer Center at the University of Oklahoma in Oklahoma City. And of course, I'm Rob Coleman, GYN oncologist here in Houston, Texas, where I serve as Chief Scientific Officer for U.S. Oncology Research. So today, we're going to capitalize on the previous 2 discussions. So first is really talking about the science behind PARP inhibitors, why they work, and how they were introduced into the recurrent treatment setting, then into platinum-sensitive maintenance setting, and then ultimately the big prize, being incorporated into the frontline setting as primary maintenance. And then, yes, the second podcast, we talked about toxicities and the tolerance for these therapies, and we discussed that in great detail and some strategies on how to mitigate that. So I really encourage you to go back and listen to those first 2 podcasts. You want to get some comprehensive nature about how we use these therapies. And today's podcast, what we thought we would do is focus on the future. Where are we taking this effective therapy now used in the frontline setting in patients who have not been previously treated with PARP inhibitors, and using them as primarily as a maintenance treatment? And one of the concepts that comes up, obviously, is what do we do in combination? And we tapped into a little bit of this yesterday in podcast 1, where we talked about PARP inhibitors being used in combination with bevacizumab. So the PALA-1 trial, which we demonstrated and presented, Dr. Moore presented very nicely the efficacy data. Now we have overall survival data for that trial, and we're very excited to see that. And so the question is, how do we take this further? So first, let me welcome Dr. Moore. I forgot to do that at the beginning. So welcome, and thanks for joining us.
Thanks for having me.
All right, great. So maybe we could start by maybe talking about why are PARP inhibitors of interest to be used in combination. Maybe let's just start there, and then we'll kind of get into like the frontline trials that we're going to be hoped to hear about soon.
Sure. As we've talked about the last 2 podcasts, PARP inhibitors definitely have monotherapy activity. So these are agents that, for a good proportion of high-grade serous and high-grade endometrioid tumors, work quite well. But they don't work in everyone, and they don't work as well in everyone in whom they do work. You have folks, like we alluded to in the first podcast on SOLO-1, that hopefully are cured, at 7 years, no recurrence. And then you have that same study, which was all BRCA-associated cancers, 24% of patients recurred during the 2 years of their PARP inhibitor. So clearly, despite having the molecular biomarker, something was off, and we're learning about that, the importance of the particular mutation and then the microenvironment, et cetera. We're unraveling that sort of as we speak. So I think that when we think about PARP inhibitors, we've made a lot of accomplishments, but this is the new floor, and so now we're building off of kind of where we are now. We have not peaked, and we have a lot of patients left to serve. specifically all of those who aren't cured with PARP inhibitors alone. So the interest in combinations kind of comes from that, is that basically we're not done. And then the rationale for the particular combinations really comes from our colleagues who are doing really elegant preclinical work, both in cell lines and animal models, to show how you can improve efficacy of PARP inhibitors with rational combinations, sometimes in biomarker selected subgroups. And so some examples of that we can dive into, maybe a few. We don't have time to go into all of it, but there's rationale for synergy between PARP inhibitors and anti-angiogenic agents. And that has led to a lot of development and ongoing clinical trials, and actually phase 3 trials. And it's kind of the basis for the PAOLA-1 study, amongst others. There's evidence for synergy between PARP inhibitors and agents that target the PI3 kinase pathway. And so, our good friend, my good friend, Shannon Weston, has shown beautiful synergy both with AKT inhibitors as well as mTOR inhibitors, but probably the strongest signal comes from the work of Gerberg, Wolf, and others with PI3 kinase inhibitors and PARP inhibitors, this metabolic intervention. That has moved into phase 3 studies. And then the preclinical work for PARP inhibitors and immune checkpoint inhibitors, really hinging on activation of that STING pathway to sensitize tumors to immune checkpoint, was the rationale for some other drug development. So, and there's others, a myriad HDAC inhibitors and many other things we could talk about. We could have hours of conversation about the combos. But the particular thing of interest is that many of the models in which these projects were done, PARP inhibitor and ATR is another big one. That's Dr. Fiona Simpkins' work, were done in really well done models of PARP resistant tumors that won't respond to PARP alone. And then the combination works very well, which is the kind of work you need to do these days. We should have been doing all along, but particularly now. Before you move something into clinic, like, you really have to show some proof of concept in a tumor that's of high unmet need and start to individualize. So, I think we're starting to get there. Those are just 3 of the studies, combos that have moved forward. We could talk about others.
Yeah. So I always kind of say that, when people say what's next for PARP inhibitors, or for any compound for that matter, we always say, well, one, we'd like to make them work better in patients that they already work. So how do we augment that activity? 2 would be how do we use them in ways where patients have become resistant to them? So this would be a situation where there's either kind of an acquired event of PARP resistance. And then the third would be to see if we could get them to work in cases where we wouldn't expect them to work. So these would be ones that, say, have intrinsic insensitivity. And I think you basically talked about all 3 of those kind of areas of where we want to proceed. And of course, we've got this data that has emerged out of the Topasio and out of the Mediola trials that have looked at PARP inhibitors, in combination with immune checkpoint inhibitors, and in Mediola, in addition of anti-angiogenesis inhibitors. So, and obviously, I think the signal we got from that, as opposed to, let's say, Moonstone, where we looked at this in the platinum-resistant patient setting, the platinum-sensitive, setting appears to have, potentially some strong activity there. So maybe you share with us some thoughts on, how the patient populations might dictate what might be the most exciting, opportunities to move these many different ideas forward for for a clinical trial.
Sure. And just acknowledging up front that these are sort of my opinions pending results of clinical data. So this is sort of what I think, and I could very well be wrong.
A data-free zone.
Data-free zone. This is what I think is happening. Let's start with the IO immune checkpoint inhibitor plus PARP story, which there were several studies around. You just alluded to Moonstone. We have Topasio. We have Opal. Either PARP inhibitor plus an immune checkpoint inhibitor, or PARP inhibitor, immune checkpoint inhibitor, bevacizumab in a platinum-resistant setting. And the initial studies of that, and these were run by our good friends and brilliant researchers, Drs. Panos Konstantinopoulos, Leslie Randall, and Joyce Liu, well-run studies with very modest to no signal of efficacy. mean, we have to acknowledge a response rate of barely 20% in a platinum-resistant space is really not a marker to move forward. However, at the same time those were being performed, a similar experiment was being done, and you just referenced the Mediola study, which was updated at ESGO this year by Dr. Susanna Banerjee. And this was a study done in platinum-sensitive disease instead of chemotherapy, so no platinum, and in BRCA wild-type tumors. So those that aren't guaranteed, you're never guaranteed, but you can't just say, oh, it was just the PARP, right? This is a platinum-sensitive PARP, BRCA wild-type, and they tested the olaparib durvalumab versus olaparib durvalumab and bevacizumab. And it was a small study, but they also characterized the tumors as genomic, the GIS score, which is the myriad HRD score. So GIS positive, negative, or unknown. And so in a GIS negative, that's homologous recombination deficiency test negative, you would not expect any efficacy from a PARP inhibitor. Even in a platinum sensitive HRD test negative, we know that from Quadra, the response rates 5 to 10% at best. But what we saw in Mediola was a 77% response rate. And it was like 100% response rate in the HRD test positive. And these are in the triplet, for the triplet therapy. The doublet didn't work really well, which is interesting. So is there some magic to this triplet of targeting angiogenesis, targeting PARP, and targeting PD-1 or PD-L1 in this case, in a context of a platinum-sensitive tumor that creates some synergy and some therapeutic magic? That result may be spurious. It's a very small study, or it may be real, but really we're all kind of on pins and needles to see the result of one of, really 3, but 4 frontline studies, which are all comer in terms of HRD status. 3 of those studies have that triplet in at least 50% of the population. So cumulatively, it's several thousand patients who have received the triplet versus the doublet. And so we should be able to tease out whether or not there's some magic to this this triplet therapy. But I think if it does work, if IO has a hope in ovarian, I think that's the real question. The prior failures may have been related just to the context of the tumor and being so platinum resistant that maybe it's just too late. But when you bring it in with appropriate partners in a tumor that's either less heavily pretreated or still quote unquote sensitive, whatever that means in the tumor microenvironment, maybe you see a benefit. There's a lot of companies riding on that hope right now. And we'll start to see that next year.
Yeah, and that's a good point. So, I think I like to break this down, kind of our expectations. So in platinum-resistant ovarian cancer, so these are patients that, have demonstrated that they're no longer sensitive to a DNA damaging agent as strong as platinum. So our expectations for PARP working by itself in that situation, very low. Same thing with immune checkpoint inhibitors. We know that ovarian cancer is not a mutationally loaded tumor. It's annotated by copy number alteration, relatively low mutational burden, and we wouldn't expect, and we haven't seen activity with IO therapies alone in that setting, nor in combination with chemotherapy. So we've got these two agents that really don't have any activity in that setting now being combined, demonstrating low activity in that setting. So we didn't really achieve that push. But as mentioned, in the platinum sensitive setting, where we've actually kind of teed up the patients to potentially respond, we don't really have strong signal with IO in platinum sensitive disease, but we have a strong signal with PARP in that setting. We have seen activity with bevacizumab that is not dependent upon BRCA mutation, for instance. So it has its own independent activity. So we're starting to see that these synergies may work in certain patient contexts, which is why it's so exciting to talk about these ongoing trials that you mentioned. And I'll just list them here for sake of clarity. So we have FIRST, which is combining dostarlimab as the immune checkpoint inhibitor with niraparib as the PARP inhibitor, and bevacizumab is an option. We have DUO-O, which is combining durvalumab as the immune checkpoint inhibitor with olaparib as the PARP inhibitor, and bevacizumab is given in this patient cohort. And we have Key-Link, which is the using pembrolizumab as the immune checkpoint inhibitor with olaparib as the PARP inhibitor. And here again, the bevacizumab is left up to the investigator. And the fourth trial that you mentioned was the combo, which is looking at the immune checkpoint inhibitor of nivolumab with the PARP inhibitor rucaparib. So it does not have that bevacizumab component to that. But as you mentioned, that accounts for 5,000 patients. That's a huge number of patients with 4 different immune checkpoint inhibitors, 3 different PARP inhibitors, and in the context of bevacizumab. So tell me how you're going to sort through all of that data when it comes out. Because these are being done, some of these are limiting patients that have BRCA mutation or tumors that have BRCA mutation. So these are going to be largely focused in the tumor BRCA wild type cohort. So we'll have HRD test positive patients, but it's really going to complicate the landscape if some of the results are positive and some of them aren't. So right. I think, yeah. So how are our listening audience going to be able to sort through something as complicated as 3 drugs in a maintenance setting and interpreting these trials?
I mean, honestly, I think it's just going to come down. These are all well-developed or well-designed, well-executed studies. They're very different, though, as you alluded to. I'll tell you, for example, the first study is based on the homologous recombination repair panel, not HRD. So it's a different biomarker. Mm-hmm. Key-Link is just in BRCA wild type, and Duo is kind of the same, except it has a separate cohort for BRCA. So it's really going to be breaking down the biomarkers. I think if they're all positive, the hardest to interpret is going to be my study. So I guess I can say that out loud is first, because we have a different biomarker, that's not commonly used in the United States, at least yet, as a predictive biomarker for PARP inhibitors. And so it's just going to come down to the results, how the kind of control arms, we sort of look at the control arms and sort of level set, is that a overperforming or underperforming control arm? What does the population look like as compared to the other relatively recent data? So it's fortunate that we have relatively contemporary data sets in Prima, Paula. And Solo is kind of an older study now, actually, to compare to. So it's not like we're comparing outcomes to a decade ago, where all sorts of things can impact that. So I think it's just going to come down to the results and the pre-specified analyses, analytic analyses. I think I'm super frustrated by decisions being made by non-analytic endpoints. So hopefully that stops with these studies. And also the magnitude of benefits is going to be very important here. We're talking about 3 drugs in a maintenance setting, and to show a clinical and statistical benefit in frontline, usually the magnitude of benefit's pretty high. It's hard to show a benefit there because generally patients do pretty well for a while. So the magnitude of that benefit is going to be important in whether or not providers and patients are willing to take 3 drugs.
Right, 2 IVs and an oral. I completely agree with you. We didn't talk much about two kind of really key questions that come up when we talk about these trials. One is patient diversity. We have made a very concerted effort now that the FDA has requested a diversity plan be accompanying INDs as they're filed. And secondly, financial toxicity. So both of these are going to be looked at. Why is diversity so important for these kind of frontline, well, actually any clinical trial, but especially in trials that are enrolling, now 5,000 patients answering these specific questions? What are your thoughts about diversity in clinical trials?
Well, I think finally there's really a national kind of NCI-led push to not just report. Okay, we've been in reporting mode for a long time about outcomes of all sorts of cancers. This isn't unique to gynecologic cancers, based on mainly ethnicity, but it's such an intersection between ethnicity and socioeconomic status and geographic and financial disparities. So there's this kind of intersection that comes together that historically has excluded really substantial proportions of kind of US citizens and not even, and US non-citizens, I'm going to say, from not just trials, but just access to NCCN level care at least. So that's sort of the first problem, right? Is just making sure that everyone has equitable access to exceptional care. And then as a part of that, clinical research is clinical care. And so you get access to trials. And so I think we've just been in this reporting mode of, Black men with prostate cancer do far worse than white men. And, indigenous populations with cervical cancer do worse than, white women. Okay, we've been saying that for a long time, but we're finally doing some implementation science around it to overcome those disparities. And I really feel like it was forced on us, not forced because of the importance of this, but the changes were forced on us by the changes that were made during COVID, many of which probably unintentionally led to more equity in access to physicians and therapies and trials, because we took out a lot of the fat that often can be cumbersome for patients to participate, to even receive therapy at an academic center or participate on a trial. Virtual visits, I think, were phenomenal for opening the doors, allowing patients to treat with standard medicines closer to home when they're on a trial was phenomenal. Direct delivery of medications to patients. Instead of having them come to Oklahoma City, they can just get their medicine shipped to them. And it kind of leveled the field for a lot of our patients with financial toxicities and geographic disparities, which does encompass a substantial number of patients who are ethnically diverse. So I feel like we sort of showed that we could do this better and be more equitable in access to treatment and trials with COVID. And that has been picked up by the NCI and is now, as you alluded to, really a big part of a lot of the diversity plans, just to overcome a lot of the barriers that keep a lot of patients from accessing trials. And why is that important? Well, I guess I don't know why we shouldn't have to explain this. It's important because, trials save lives, and you and I see this every day. They don't always work, and that's just the truth. But sometimes they work, and they work really, really well. And nothing makes me happier than to come to work. I have a patient who's from one of our sovereign nations here in Oklahoma City who has, we're talking about ovary, but she has uterine papillary serous cancer, and she's HER2 low. But that now qualifies for a study. And she's on a study right now with what should have been a terminal cancer, and she's in a sustained partial response and doing great because she's on a trial. It's just like, it makes me like giddy to think about it, that she wouldn't have had access to anywhere else, but she's got to travel all the way here from her community. So there's still things to overcome. But now we're going to have data on how these medicines work in people who are from sovereign nations, who are oftentimes completely like zero participants of our indigenous peoples on any trial, maybe one. That's a problem because maybe the drug doesn't work as well, or maybe the disease is different. Same with, Black women or just Black people in general. Endometrial cancer is a great example, again, where endometrial cancer is one of the few tumors that's increasing in incidence and mortality, largely impacting Black women, and they're absent from the trials. And so does the drug work as well? Is it as toxic? Should the dosing be different? How do we best take care of the patients who are actually dying of the disease? And so ovarian cancer is no exception. You had some data up, and I've seen it as well. We're in this sort of weird, in a good way, conundrum where the incidence and the mortality of ovarian cancer is declining by about 30%. It was pretty nice over several decades, but not amongst Black women. It's not. It's gone up a smidge, but certainly not going down. And why is that? It's probably multifactorial, but one thing we know just from market data, a lot of market data, is that Black women aren't being offered genetic testing at the same rates as white women. They're not being offered it till later when they're a very advanced disease. So early stage diagnosis, we're not testing. And they're not getting PARP inhibitor maintenance offered to them at the same rates as white women. And we know that that has transformed, we think, OS, or based on early data out of Solo and Paula, but at least PFS. And we know that they're not being offered these medicines. And so
Right.
how is that even remotely acceptable? It's not. So we need to continue to push this kind of message forward and do all the things that people are doing to basically require that you overcome identifiable barriers to allow all patients to have access to a trial, and they can choose whether or not they want to participate, but it shouldn't be made for them by extraneous considerations.
Yeah, that was really well put. I think, the solution to this disparity that we see is complex, very individualized. But I think that we are trying to make efforts to do that. One of the things that has come up has been that on a global scale, trials that are run globally, is that there has been an attempt to reach into ethnically very homogeneous populations in other countries, and I think that what we learned from that is that what we're trying to do is to represent diverse populations where there's common exposure within the same country. So for instance, an Asian American who's lived their life all in the U.S. may be different than, say, an Asian patient coming from rural China. And so we're trying to understand these pharmacogenomic differences in these patient populations, and that's why it's been so important. We also, we try to match the diverse patient population to the disease category. So as you mentioned, Katie, we see African American women with high rates of high-grade histologies and endometrial cancer. So when we write trials for that setting, we're trying to represent that population. Ultimately, the goal is that when we produce a trial result in these ongoing studies, that it's applicable to these drugs being used, under less stringent circumstances outside of a clinical trial, and so that we don't run into unexpected toxicities or unexpected lack of efficacy, that they mirror that patient population. So that's why we feel that this is such an incredibly important component of it. And as I mentioned, almost all of these trials now have ongoing monitoring of diverse population participation. So great. And for all those of you who are listening, if you have access to clinical trials, please, please, please support them. This is, as we mentioned beginning, this is how we move the needle forward. It really is the standard of care. And so I hope if you have it at your disposal that you'll support those trials. So Katie, let me just wrap this up. We've had a tremendous amount of information given. Thank you so much for your expertise and your commitment to continuing the clinical trial effort in GYN cancers. Today we're talking about PARP and ovarian cancer. Obviously, we're very excited about that work that you've done and continue to do. We are very excited about the future, looking at ways that we can extend the use of these agents and make them even more effective. We have a lot of really cool ideas that you brought up about what's coming in the trials that have already enrolled, but also some new opportunities looking at different pathways and ways to leverage these medications. So I want to close out by thanking the listeners as well for your participation in these podcasts. Katie, thank you again for your tremendous knowledge and the clarity. I think that you've brought this message to our audience. It's really a gift that you have, and I really want to extend my gratitude to you for joining us for these 3 podcasts. So thank you so much.
Thank you for having me.
You bet.
Remember to receive your credit and evaluate this program. Please visit morningcommutepodcast.com/ovarian cancer 3. You can find all of the episodes in this series and all of our other podcasts on your favorite podcast streaming services, or download our Morning Commute app. Thank you for joining us today.